Thank you for having me here today. I've been here at Mahidol for 1 week, and I'm very happy to join your faculty. And I really like the campus, and everyone I've met has been very friendly. So I look forward to working with all of you. My name is Thomas Stewart, but everyone please call me Tom. And I have been working in the United States for the past 30 years, predominantly developing new technology for environmental analysis. And I have a lot of stories I could tell you, but we only have a short amount of time today. So I'm just going to give you a brief overview of my career and talk a little bit about what my current plans are for research. I have a PhD in biological chemistry from the University of North Carolina. Following my education, I spent 5 years with a company called INSYS, where I learned to make environmental diagnostic test kits. After 5 years at INSYS, I started my own company called Mercury Science. That was in 1995, and I've spent the past 22 years as president of Mercury Science. Mercury Science is focused on developing new methods of analysis for soil, water, and food. I have a variety of experiences, or I should say skills. So in developing the environmental test kits, you need, or I have experience in analytical chemistry, organic chemistry, biological chemistry, and environmental chemistry. And my work has focused on subjects such as detection of heavy metals such as mercury, pesticides, hydrocarbons like petroleum products like gas, PCB, polychlorinated biphenyls, dioxins, and toxins in food and water. My company is located in North Carolina, which is this part here of the United States. The company focuses on new analytical methods development, and we have done our own research, but we also do contract research and scientific consulting for other labs. So we work with small business labs, academic labs, and government labs. So when these labs need a special service that they don't have the technology in their own lab, they'll come to Mercury Science and we will do the work for them. We also manufacture and sell our own products, and our two best products are the Mercury Spot Kit, which detects mercury in soil, and a test kit called domoic acid test kit for detecting a shellfish toxin. Some of my past projects have included detecting mercury in soil, detecting dioxin in incinerator waste, detecting polychlorinated biphenyls in pig fat, and detecting melamine in milk. Mercury Science has had collaborations with a variety of organizations, including national departments such as the U.S. Food and Drug Administration, the National Oceanographic and Atmospheric Administration, which handles the quality, the water and air quality of the United States. I've worked with state labs such as the Department of Health in California and Alaska. I have a lot of projects that I've completed with university labs, and of course I always work with other industries and companies. A large focus of my work has been on method development, and as a small company, you cannot develop new technology on your own. It's a team effort, and this slide just depicts a example of a project and the number of different groups that were involved in the development effort. So it includes working with private companies, in this case a company in Raleigh, North Carolina. Here's an Alaska department, the NOAA department. Here's a government lab in Seattle, Washington. We had Native American tribes using these products and interested in collaborating with us, and university labs. And all of these groups together work together to make a new test method to detect toxins in shellfish, which we then submit for methods approval. In this case, it was the International Shellfish Sanitation Conference. I've been making these environmental test kits for almost 30 years now, and one question is, why spend so much time making better test methods? And there's been a lot of change in the world in the last 30 years. The world is more connected now. We're sharing resources. We're sharing food. We're sharing other products other than food. There's a lot of global interactions, and we need to know what we're bringing into our country and what we're exporting from our country. So, for example, two projects that I've worked on. One was for melamine in dog food. Several years ago, the U.S. was importing dog food from China that was contaminated with melamine. Melamine was a toxin which was making our dogs and cats sick and dying. And I worked with government agencies to develop a test kit for melamine in dog food and milk. And that is an example of importing a food into the U.S. And we had another example of exporting from U.S. to China. We were exporting shellfish, and China found toxins in the shellfish and banned the importation of these shellfish. So we developed a test to detect the toxin in shellfish. Another reason we need to improve these test methods is to ensure public health is safe with clean drinking water and safe food. 30 years ago, when I started in this field, environmental analysis was expensive and complicated, and samples needed to be shipped to a lab, taking between 1 and 3 days from when the sample was shipped to the lab until you got a result. And in the modern world, we can't wait 3 days for test results. We need test results now. So the focus of my work is in making tests faster and simpler than the previous generation. Another reason to have better test methods is to monitor our factories that are producing waste to help protect the environment, save endangered animals and plants, and preserve the beauty and health of our ecosystems. This slide shows a little bit about what we are doing to make better methods of environmental analysis. And basically, environmental analysis is following along in the path of medical diagnostics. So we spend a lot of time and money developing medical diagnostic assays to help our patients in our hospitals. And the environmental industry has been taking advantage of this work by converting medical diagnostic breakthroughs into environmental methods. So before 1990, a traditional analysis took place in the lab with a GC or an HPLC, and it required expensive equipment and extensive training. It took a lot of money and a lot of time to get a result. But starting in 1990, we had the first use of medical diagnostic technology in an environmental test kit. And this test kit used antibodies, which before 1990, environmental analysis did not have. So we took antibodies to, for example, in 1990, our first test was for PCB and soil. And we could analyze the soil for on-site for the presence of PCBs. And this test took only 1 hour, and it was cheaper than the GC test, but it was only qualitative, not quantitative. By 1995, the home pregnancy tests were common in the medical device field, and Mercury Science took a home pregnancy test and modified it to detect mercury in soil. So here's an example of a positive sample. When mercury is present, you'd see the letters Hg form on a white membrane. And now, this year in 2017, we've moved on to modifying blood glucose meters and the electrodes to detect toxins in the environment. The advantage of these blood glucose meter technology is that we can do a test in about 1 minute, it's very cheap, and it's quantitative. If anyone has any questions after my talk and would like to come up and speak with me, I'll be glad to talk in more detail about these things. One thing I brought with me to show you today is a typical blood glucose meter available in the United States. And this little device costs about $20, and you can take a drop of your blood and measure the amount of glucose in your blood in just about 5 seconds. And I'll explain more how it relates to my work in a little bit. Here's a closer look at the example of the first product that Mercury Science developed. It was a test to detect mercury in soil. And as I mentioned earlier, it's very similar to a home pregnancy test. If a soil did not have mercury, you would have a clear white background on your assay device. But if there was mercury in soil, you would get a blue color. Very easy to tell whether it was a positive or a negative result. Here's a better picture that everybody can see of a typical blood glucose meter. And they're very cheap. They have electrodes that you use once and then throw away. These are very cheap electrodes. We call them single-use disposable electrodes. And one thing to keep in mind is that this meter is actually a potentiostat, and the test strip is actually an electrode. And the product takes advantage of electrochemistry to measure the glucose in a blood. Electrochemistry provides a fast, simple, and quantitative means of testing a sample on site. Mercury Science has taken that blood glucose meter and modified it so that we can detect toxins or heavy metals, chemicals in soil, food, and water. This is the prototype potentiostat that is adapted for use in the field. It's battery powered, simple to use, inexpensive, and is able to quantitatively measure your analytes. This device here is the electrode, similar to the electrode seen in the blood glucose meter. And I have an example here to show you. So I brought with me today the prototype potentiostat that Mercury Science has developed. And this device can be taken out on a boat, on the beach, out in a rice farm, and we can do the analyses right there instead of having to send samples back to a lab for analysis. The technology takes advantage of screen-printed carbon ink electrodes, and these can be mass-produced simply and cheaply, and we can modify the surface of the electrode to detect the analyte of interest that we have. Here's a close-up of the sensors that we've developed. It's basically has 4 electrodes. We have these 2 ovals here, which we call working electrodes or sample electrodes. Oh, I'm sorry, these arrows are wrong. For some reason, the slide is not correct. So follow the red light. We have here a sample electrode, a control electrode. The silver or gray electrode is a reference electrode, and this surrounding zone is a counter electrode. I apologize for the mistake on that slide. We're able to modify the surface of the electrode. In this example, here's the carbon ink electrode, and we put a layer of antibody on, attach a layer of antibody onto the electrode. This antibody is specific for any analyte that we want to choose. In this case, we're detecting domoic acid in a shellfish sample. So the antibody binds to domoic acid, and we do a competition between the analyte in the sample and a tracer, which is domoic acid conjugated to an enzyme called horseradish peroxidase. Let me go back and just mention that there's a competition for the antibody binding site between these two competitors. And what we want to measure is we want to measure the amount of domoic acid, HRP, which binds to the electrode surface. Once the HRP is mobilized on the electrode surface, we add a substrate which contains TMB and hydrogen peroxide, and the HRP catalyzes the oxidation of the TMB on the electrode surface. We apply a potential to the electrode, which reduces the oxidized TMB, and we create a current. And that current that we create is proportional to the amount of HRP that's captured on the electrode surface. We can run assays with standards containing known concentrations of domoic acid and measure the current that results at each concentration and develop a dose-response curve. Once we know that dose-response curve, we can run an unknown sample and measure the current and compare it to the standard curve to obtain the concentration of domoic acid in the sample. What are the advantages of the electrode biosensor versus a traditional lab test? The potentiostat is small and battery powered, so it makes it portable. We can use it outside the lab, take it anywhere to do our testing. The test is simple to run so that anyone can do it. You don't need a college degree to be able to measure your samples. The test is rapid, takes less than 1 minute to get a result, and the test is inexpensive, so we can do a lot of samples in a short amount of time, and it's not cost prohibitive. So I'm glad to be working here at Mahidol, and I would like to propose that we have some plan on how to continue my research here at Mahidol. I would like to meet with all of you that are interested and identify some analytes of interest in the environment that are causing some problems possibly in Thailand, such as maybe pesticides or microbiological organisms or heavy metals, industrial waste, seafood, anything that the department can come up with. Feel free to come and ask me if my technology might be helpful for your area of interest. I'd like to build a multidisciplinary team. I cannot do this research by myself, but together as a team we can accomplish great things. So for my technology, we need chemists and biology majors and engineers, people to write software and create the hardware for my instruments. And it would be nice. I'm used to working with both academic labs and companies and government agencies to collectively learn where the problems are, what the problems are, and how we want to solve problems. Once we know what we want to develop, we can conduct research and development projects. and develop prototype assays and demonstrate usefulness of these assays. And during this research and development, we're also teaching and training students how to do this research. If we're successful, we can patent any technology that we develop and publish our research. And everybody benefits from this. And if the technology is good enough, we can consider commercializing new products, starting a new company, manufacturing, selling products in both Thailand as well as Asia and even the global market. That's all the science I'm going to talk about today, but I'm new here and I want to meet people and have friends, so I want to tell you a little bit about myself. I like to play sports, so if anybody in the department likes to play golf or tennis or badminton, let me know. I'd be glad to play. I'm learning to read Thai. So right now, to learn to read Thai, I'm enjoying reading Pet Prat Ma, and I've read 25 books already and still working on them now. I like to take nature walks, and I take a camera with me, and I like to photograph things such as butterflies and birds. Anything else I see that I think strikes my eye. That's all I'd like to talk about today, but please feel free to come up and ask me any questions if you'd like, or I'll answer any questions you may have. Thank you. Oh, here's my email address if you'd like to contact me by email. You can also contact me at my company email address here, or you can connect with me on Facebook at tom.stewart.140 is my name on Facebook. Thank you.